Hi, I'm Van Lê. I'm a rising senior at Hollins University studying mathematics and applied economics with a concentration in data analytics. This summer, I'm working on the project Discovery of Heterogeneous Treatment Effects from Landmark Clinical Trials for Cardiovascular Disease. My mentors are Chirag Napar and Professor Arthur Dubrowski. As of now, there are more than 400,000 randomized clinical trials conducted worldwide, and in the US alone, each year, the National Institute of Health spends about $41 billion in medical research, and these clinical trials play a very important role in research, development of new drugs, and implementation of new treatments to patients. However, the 400,000 is just a fraction of million trials not registered, not published after concluding that the treatment is ineffective. The treatment effect, however, may vary because of the differences in the response characteristics to the interventions of different patients. And this characteristic can be demographics or phenotypic health characteristic or medical history of the patient. However, the presence of treatment effect heterogeneity is often excluded in the evaluation process of randomized clinical trials. So in this project, we utilize the existing deep Cox mixtures with heterogeneous effects model to study the heterogeneous treatment effects in 3 landmark randomized clinical trials, including prevention of events with angiotensin converting enzyme inhibition trial, action to control cardiovascular risk in diabetes lipid study, and lipid lowering treatment to prevent heart attack trial. And this project shows that the examined critical trials were more efficient to certain subgroups of patients, which we call pheno groups, and this one demonstrates the importance of the discovery of heterogeneous treatment effects, and this will be beneficial to the stakeholders, including the doctors, policymakers, when they decide on the type of treatment that can be customized for each patient. So the model that we used in this project is the CMHE model of the open source package called Alton Survival. And the efficacy of this model was demonstrated in its original paper by evaluating the performance on 2 landmark randomized clinical trials, which are antihypertensive treatment to prevent heart attack trial and action to control cardiovascular risk in diabetes glycemia trial. And in those 2 studies, the authors of the model has shown 2 different phenogroups which benefited differently from each intervention in the clinical trials. So I'm going to explain the methodology of the project. So we start with pre-process the data, which are the features of patients recorded at the baseline of each trial. Data pre-processing is one of the most important parts of this project because the accuracy of our model heavily depends on the accurate transformation of raw data into understandable formats. The features were separated into numerical and categorical features and were processed with a built-in class from the Alton Survival package called preprocessor. After the data was pre-processed, we instantiated the model with the appropriate set of hyperparameters and fitted it on NumPy arrays, consisting of the pre-processed feature data, event or censoring times, and the treatment. After this step, we got the estimated latent treatment effect variable phi given the confounding features. This latent group phi mediated the treatment effect, and at this step, each patient is assigned 2 probabilities of treatment effect corresponding to the 2 interventions, treatment or control. And if the patient has higher probability of event-free survival in the control group, he or she will be assigned to the harm group. Otherwise, he or she will be assigned to the benefited groups. We also got the hazard ratio and the 95% confidence interval of hazard ratio for each group. The hazard ratio is equivalent to the odds that an individual in the group with the higher hazard reaches the endpoint, which is the primary outcome of the clinical trial. In other words, the higher the hazard ratio, the higher chance of the treated patients healing first. Finally, we apply the decision tree classifier models on the data to get the features that affected treatment outcomes. The numerical data used in this step are data before normalization so that it is interpretable to stakeholders. After the experiments, we found that there are clearly statistically significant differences between the survival rate of patients in each intervention group, which we called phenotypes, harm groups or benefited groups. And we can see that, for example, in PEACE trial, the decision tree classifier reveals that the patients who have history of smoking and lower baseline blood pressure would benefit more from the Trendelenburg treatment. And in the on-head lipid study, features such as low total cholesterol and high weight would make the patients more suitable to be treated with pravastatin, while patients with high baseline, high-density lipoprotein cholesterol and lower blood pressure should be given usual care. Similarly, in our core lipid therapy trial, we also revealed two phenol groups with diminished or enhanced treatment effects from the treatment lipid fibrate or the lipid plausible. We hope that more research into major clinical trials will be conducted so that the application of the CMHE model will be more beneficial to the development of the customized treatments to patients. Thank you.